Yes, thank you. My name is John Reynolds. I'm a professor here at Salk, and I'd like to welcome you to the 20th anniversary of the Kavli Institute for Brain and Mind. And today we're gathering to explore a fundamental theme that is emerging in neuroscience, which is the construction of internal models that guide perception and behavior. This is a concept that has very deep roots, dating at least back to the 11th century in the work of Ibn al-Haytham, a wonderful Arab scholar who was the first to appreciate that perception is not merely passive sensation, but rather is a process of inference based on our knowledge of the world. And this concept has carried powerfully through psychology over the years, exemplified, for example, by the work of the great late Richard Gregory, who created wonderful illusions to demonstrate how our perception is really governed by our appreciation of the statistics of the environment. And for those of you who haven't seen it, I recommend you, after the meeting, go look up the Charlie Chaplin mask illusion, which is one of his best and really worth listening to. Today, this idea of internal models has really reached a fever pitch in artificial intelligence. Large language models extract the statistical regularities of the natural language, and they can then be used to generate entirely novel utterances in the language, sentences, paragraphs, entire books that are close to human quality in some cases. And our symposium brings together 7 distinguished speakers from diverse backgrounds, including philosophy, neuroscience, and artificial intelligence, to explore these themes. And it's my real pleasure to introduce our first speaker, Doris Tsao, whose work really exemplifies this theme quite beautifully. She is an HHMI investigator at Berkeley, and she was recently awarded the Kavli Prize with Nancy Kanwisher and Wienrich Freiwald for their work developing our understanding of face perception in the brain. And as she will tell you, she really discovered a patchy network of areas in the temporal lobe that interact to create internal representations of faces. And this is really profoundly important work, and it really may be a sort of Rosetta Stone for the coding of information in the world, because she's now undertaking work looking at how these different multiple brain areas that she's discovered interact with one another, much like a large language model would generate a sentence, but given some data from the retina, can construct an internal representation of a face that you'll recognize, can see in 3 dimensions, and interpret its emotional state and so forth. And so without further ado, I welcome Doris Tsao, and thank you all for being here. I will make one practical point. There's a microphone here, so if you have questions at the end of the session, please come to the microphone. Thank you.
Thank you so much, John, and to the organizers for inviting me. It's really a delight to be here. And it's wonderful to celebrate the 20th anniversary of the Kavli Institute for Brain and Mind together with you. So I want to start with a personal story about how I became a visual neuroscientist. And I remember the day very clearly. I was a sophomore in college, and my dad came to visit me, and he showed me a paper on 3D vision that he had written with this equation in it, and I thought this equation was unbelievably beautiful. So what the equation is expressing is the following simple idea. If you look at a patch of the world from 2 different perspectives, the image patches will be transforms of each other. And a central challenge of 3D vision is to compute this transform. And my dad's idea was to equip receptive fields with the capacity to warp and stretch so they can generate the transform caused by perspective change, and thereby compensate for this transform. And I found this idea that the brain might have receptive fields that are actively generating transformations in order to perceive the world so beautiful. And since college, my dad and I have been working on a paper developing this idea, and we finally published it 2 years ago. And I tell this story to convey that from the very beginning of my interest in vision, I've been deeply drawn to the idea that the brain has machinery to actively generate perception of the world. So, which brings me to the central topic I want to talk about today, the brain's generative model for reality. So in my talk, I first want to introduce the concept of a generative model. Then I'll introduce you to the experimental arena in which we'll tackle the question of whether the brain uses a generative model for visual perception, the macaque face patch system that John just mentioned. And then I want to tell you 4 stories about the neural code for facial identity, representation of degraded faces, neural dynamics during binocular rivalry, and finally, responses in human IT cortex during visual imagery. And I'm going to argue that the primate brain does possess a generative model, and each of these stories provides a different lens into the nature of this model. And finally, I'll conclude with some general thoughts. So what is a generative model, and why do we care? I think of a generative model as something like a video game engine, a set of precise knobs controlling the existence and properties of every single object in a scene. So when visual input first enters the eye, it's just a cacophony of pixels, but through a series of stages of visual processing, it becomes organized into a sensible representation that carves nature at its joints, so to speak, and identifies the generative factors responsible for building the scene. And I think it's obvious that this is desirable, so you can know, for example, a ball is coming at you, and that's what you should be worried about, not the umbrella or the trees. But if you're feeling hot, then you might want to go move the umbrella. And so having this type of explicit high-level model is what it means to understand the world. So how does the brain build a generative model of the world? We still don't know the answer, but there's one idea that's been hugely inspiring to many vision scientists, including the fellow speakers today, I think, because it feels like it has all the right ingredients. And this is the idea of analysis by synthesis. And Jeff Hinton describes this idea beautifully in a paper from 1995, where he writes, "When there's no external teaching signal to be matched, some other goal is required to force the hidden units to extract underlying structure. In the wake-sleep algorithm, the goal is to learn representations that are economical to describe, but allow the input to be reconstructed accurately." And the wake-sleep algorithm is one specific generative model that Hinton proposed. So the key idea is that you can extract underlying structure of the world by learning representations that are economical to describe, but allow the input to be reconstructed accurately. So let me unpack this. According to the idea of analysis by synthesis, when an image first comes in, it passes through a series of hierarchically arranged processing stages in a feed-forward direction to build a compressed model of the input. But we're not done. The analysis by synthesis framework says that the visual system should then try to synthesize the image predicted from the initial interpretation through feedback connections. So for example, when we see a face in this image, then all the features would be regenerated from the compressed high-level model of the face through feedback. So you can think of this compressed model as something like a block of ramen, and we add water to it through the rendering process that's acting through feedback to regenerate the original image. And the step of regeneration is critical because this is what lets the brain decide if the compressed latent representation is actually valid, by checking if the image that's reconstructed from this compressed representation actually matches the incoming image. So again, the key idea of analysis by synthesis is that you want to learn a model of the world that is economical to describe, but nevertheless supports accurate reconstruction. And to put the scale of economy that can be achieved in perspective, I'm sure all of you have played with DALL-E or Stable Diffusion, and these are generative AI models that let you create images using text captions. And a remarkable thing is that the size of the Stable Diffusion model, which lets you create a 512 by 512 image of virtually anything you can possibly imagine, is just 4.2 gigabytes. And this is vastly smaller than the size of all possible 512 by 512 images. So Stable Diffusion is taking a text prompt and generating an image. And the idea of analysis by synthesis is that the brain is doing the reverse, taking the incoming image and extracting a representation that's hugely compressed, yet is still rich enough to reproduce the input exactly. So here, it would certainly need to be slightly richer description than just cute, magical flying dog. The key insight here is that the world of physical reality is much lower dimensional than the space of all possible images. And we just want the brain to accurately and explicitly model physical reality. So besides explaining how the brain can learn an extremely efficient representation in an unsupervised way, the analysis by synthesis framework has several other powerful features. First, it provides a solution to the binding problem, enabling the brain to correctly access features of the same object that are represented across widely separated areas. Because through the generative model, the high-level representation of an object can be selected and used to sequentially regenerate features of the object that are represented across widely separated brain areas, like this for the apple and this for the banana. Second, it provides a powerful mechanism for the visual system to increase robustness by removing noise from images, because the generative process steers the brain to produce the most likely image given a high-level interpretation, which is going to be noise-free. And finally, generative feedback provides a concrete mechanism for mental imagery, hallucinations, dreams, and other psychological processes that have been suggested to relate to our creative abilities. As a student of visual perception, the argument in favor of analysis by synthesis that I personally find the most compelling is actually the next one. It's the only framework I know of that can explain the inexorable consistency of conscious perception. So when context is missing in ambiguous images, our perception is somehow able to switch between the different possible interpretations. And remarkably, this switching happens in a completely consistent way across different levels of feature representation. So if you see this as a frontal face, then you also see this piece of the edge as owned by an occluder on the right. Well, if you see it as a profile face, then the same edge is now perceived as being owned by the face on the left. So the visual system is somehow able to enforce consistency of interpretation across different levels of feature representation. And how this works is a deep mystery. And analysis by synthesis explains this beautifully. So when we see a face in this ambiguous image, all the features would be regenerated in the context of the high-level interpretation of a face, including all the local figure-ground relationships that are likely represented in an early retinotopic area. While if we see a tree and some birds in the same image, then all the features would be regenerated anew in the context of the high-level interpretation of a tree and some birds. So feedback ensures that our interpretation of the world is consistent across all levels. And if we assume that what we consciously perceive is due entirely to this regenerative process acting through feedback, then this would beautifully explain why our conscious perception is always consistent across different levels of feature representation, because it's generated to be so. So finally, I want to say a few words about nomenclature. So the idea of analysis by synthesis has a long history, as John mentioned, and it's known by different names. So there's analysis by synthesis, which is my favorite term, because it's self-explanatory. And this term emphasizes how this closed-loop scheme enables the brain to dynamically discover the optimal model. The term hierarchical generative model emphasizes the hierarchical feedback pathway that's regenerating the input. Predictive coding emphasizes how this scheme lets you represent information parsimoniously by compressing it down to the high-level latent representation. And it also highlights that the scheme can operate over time, as, for example, when ChatGPT is predicting the next word. And finally, autoencoder is the machine learning term for a network that reconstructs its input via a compressed latent layer. So now I'd like to introduce you to the experimental arena where we'll investigate analysis by synthesis, the macaque face patch system. So the macaque face patch system is truly an amazing gift from Mother Nature to systems neuroscience. So here's a side view of the macaque brain. Visual object identity is processed by the ventral visual pathway, which starts in V1, where there's a bank of filters for various local features like orientation. And then it proceeds to mid-level areas, V2, V3, V4, which process intermediate features like texture and figure-ground relationships. And finally, the representation of high-level object identity happens in a part of the macaque brain called inferotemporal, or IT cortex. And my lab has done a lot of work in IT cortex. And back when I was a grad student, I found that IT cortex contains 6 face-selective regions, which each contain very high concentrations of face-selective cells. And early on, we learned that the 6 patches are strongly and specifically connected to each other through both feed-forward and feedback connections. And this was exciting because it meant that these patches weren't just random islands of face-selective cortex, but instead they form an interacting system, potentially giving us a whole sequence of nodes to target to probe how the representation of a face is successively built up in IT cortex. And importantly, the fact that the system is connected bidirectionally makes it a promising system to look for evidence for hierarchical generative feedback. Each face patch forms a different step in face processing, and in particular, we found that cells are increasingly view invariant as you go anterior. So the middle face patch, single cells respond to faces at a specific view, like this neuron, while in the most anterior patch, responses are more abstract, and cells respond across all views, like this neuron. And overall, this face patch network provides us with a powerful template for understanding object representation that can be exploited in multiple ways. And in particular, the extraordinary concentration of face cells within face patches presented an exciting opportunity to study in detail the feature code used by the brain to represent this ineffable thing called facial identity. So can we leverage our access to face cells to understand how facial identity is represented? And that's the first story I want to tell you. And this work was spearheaded by former postdoc Stephen Le Cheng, who now heads his own group at the Shanghai Institute of Neuroscience. And to foreshadow what I'm going to tell you, we're going to see that the feature code used by face patches to represent facial identity turns out to be ideal for implementing a generative model. So let me begin by defining feature code more precisely. The feature code is the mapping or dictionary between the stimulus and responses of neurons. If we understand this mapping, then we should be able to predict the response of neurons to an arbitrary stimulus. And if the mapping is faithful, then we should even be able to reconstruct the stimulus from the responses of neurons. And if both of these were possible, then it would really show that we know the code, the language that the neurons are using to represent faces. So to figure out the brain's feature code, we first needed a way to parameterize faces. I describe each face by a set of numbers. And we considered various computer vision models for doing this, and I'm going to tell you what we found with one of the models called the shape appearance model. And this model describes every face by a set of 50 numbers. The first 25 numbers describe the shape of the face, and so here's a face undergoing changes in these shape parameters, which capture the location of key landmarks. And the second 25 numbers describe the so-called appearance of the face. So here's a movie of a face undergoing changes only in the appearance parameters. You can see they capture the texture map of the face, like the complexion and so on. So here the landmarks are fixed, but the texture map is changing. And so this shape appearance model creates a 50D space of faces, where every point is a face. And now that we have a way of parameterizing faces, we can generate a stimulus set to probe how the brain represents facial identity. And in our stimulus set, we randomly drew 2,000 faces from this 50D face space and presented them to the monkey while recording from 2 face patches. And importantly, we showed the same 2,000 faces for every cell so that we could systematically understand how each face is encoded by a population of neurons. As a starting point, we can plot the tuning of cells to each of the 50 shape and appearance parameters. And here you can see tuning curves of 2 example cells to the first 3 shape parameters and the first 3 appearance parameters. Cell 1 was significantly tuned to this shape parameter, and cell 2 to these 2 appearance parameters. So notice that all 3 curves here are ramp-shaped with a minimum at one extreme and a maximum at the opposite extreme. And this was true across the population. And these ramp-shaped tuning functions mean that each cell is acting like a ruler, measuring a face along a subset of dimensions. And mathematically, if a face cell has ramp-shaped tuning to different features, this suggests that its response can be roughly approximated by a linear combination of these features. And this has an extremely powerful consequence. Namely, for a population of face cells, we can write the population response vector as a linear transform of the 50 face features. And then we can simply invert this equation to predict the 50 face features from the population response vector by a simple linear transform. In other words, we should be able to reconstruct faces from neural activity using a simple linear transform. Is this true? We next trained a decoder to do exactly this. So we used responses to all but one of the 2,000 randomly selected faces to learn this weight matrix, and then we used the response to the left-out face to reconstruct the 50 face features with the learned weight matrix. So how good is this reconstruction? Here's an actual face that was presented to the monkey, and here's the prediction from linear regression using activity of just 205 face cells. And here are 3 more examples. So you can see the reconstructed faces match the actual faces almost exactly. So we can reconstruct the stimulus from the neural response. And moreover, from data I haven't shown you, it turns out that we can also predict responses using the shape appearance model, explaining around 80% of the explainable variance of the cells to these stimuli. And our ability to go both forwards and backwards suggests that we really know the brain's code for this ineffable thing called facial identity now. A simple geometric interpretation of the feature code used by a face cell is that cells are projecting incoming faces onto a specific preferred axis, because dot product indicates projection. And this geometric picture implies that a face cell should respond exactly the same to all faces in the plane perpendicular to the preferred axis. And this highly surprising prediction hands us a strong test of whether our understanding is correct, and the code used by face cells really is an axis code. And in fact, the prediction turned out to be true. So here are responses of a face cell to the 2,000 faces projected onto the cell's preferred axis on the x-axis and an orthogonal axis on the y-axis. And each dot represents a response to one face. Red indicates high firing, blue indicates low firing. And these 3 red dots represent responses to these 3 identities. So these 3 faces look like very different people, but they elicit the same strong response in the cell because they each have the same value of projection onto the preferred axis. And the difference between them lies along this dimension that's perpendicular to the preferred axis. And without understanding the axis code for facial identity, it would have been entirely non-obvious what linked these 3 faces. So why does the brain represent faces by a shape appearance axis code? I think there's a beautiful explanation. So recall the key idea of analysis by synthesis that we discussed at the beginning, that the central goal of visual perception is to extract a generative model of sensory input that is as parsimonious as possible. The shape appearance axis code is precisely a highly parsimonious generative model for faces that represents any face by just 50 numbers that are sufficient to reconstruct the face. And indeed, if we compare the shape appearance model to a different model for parameterizing faces that's not a generative model, such as BGG face, which is a deep network trained simply to recognize different faces, we find that the shape appearance model does a much better job at explaining macaque face patch activity, because the code used by faces is not optimized only for face recognition, but it's optimized to reconstruct faces useful for any task. So to summarize what we've learned about the brain's code for faces, cells throughout the face patch system represent faces using a shape appearance axis code, and this is a parsimonious generative model for faces consistent with the analysis by synthesis framework. And furthermore, we subsequently found that this strategy of encoding faces actually generalizes to all of IT cortex. And IT cells in general are using axes to encode objects, but now these axes live in a general object space that you can get by simply passing a large number of objects through a deep network, producing a highly compressed representation of each object in deep layers, and then you can do PCA to further reduce the dimensionality of this space. And using this object axis code, we can reconstruct general objects with responses from small numbers of cells. And so the code for objects in general is also consistent with a generative model that carries a highly parsimonious but complete representation of objects. And the first two dimensions of the object space derived from the deep network span a 2D space. And amazingly, we found a precise correspondence between the layout of this 2D object space and the topographic layout of IT cortex, where each quadrant of this object space gets mapped onto its own network of patches in IT cortex. And faces occupy one of these quadrants. So the organization of the face patch system generalizes completely to the rest of IT cortex, suggesting that whatever we figure out about face processing is likely to be true in general of all object processing. So the results I've shown you so far clarify what's achieved by the feed-forward pathway in IT cortex. And now I want to tell you two stories where generative feedback plays an important role. And we've taken two specific approaches to explore generative feedback. First, we're studying perception of degraded faces, and here the hypothesis is that generative feedback regenerates a noise-free representation that reflects the high-level interpretation. And second, we're studying binocular rivalry, where for the same visual input, conscious perception flips between two different interpretations, suggesting a generative pathway that's alternately generating one of the two possible interpretations. And so for both paradigms, we're recording simultaneously from face patches at different hierarchical levels using the NHP neuropixels probe that lets us record activity of large populations on single trials. For example, here's a raster plot of responses of over 1,000 simultaneously recorded cells from face patches ML and AF to the onset of a face. And here's the visual selectivity of a population of simultaneously recorded neurons from two face patches, confirming that we accurately targeted these patches. And it used to take us more than a year to record this much data, but now we can do so in a single trial. So first, let's discuss representation of degraded faces. And these experiments were carried out by Yuelin Shi, Franklin Franke, and Janis Hesse. So we presented to the monkey faces of 10 different individuals, each with 8 different degradations, either blur or addition of various amounts of occlusion or noise. And then we recorded with neuropixels probes from several face patches simultaneously. Here are responses of a population of simultaneously recorded cells from face patches ML and AM to this space here. So there were 398 cells in ML and 179 cells in AM. And here are responses to the exact same stimulus on 3 additional trials. So we noticed that there were sometimes 2 waves of response, as you can see here, here, and here. And each of these waves represents coordinated activity across cells and across patches. And the latency of the second wave was highly variable from trial to trial. So this is very puzzling. Here you can see example trials with 1 major wave of activity on the left and trials with multiple waves of activity on the right, where we've averaged across cells and patches for each trial. And we saw these multiple waves in about 1/3 of the trials. So what's going on here? What is the second wave for, and is it related to feedback? To investigate representational content, here we plot a similarity matrix where the x and y axis are both the list of stimuli, and the color of each entry indicates the similarity of the population response for each pair of stimuli. And the similarity matrix here was computed using responses of cells from the most anterior face patch, AM, during the first wave. And here's the similarity matrix for responses during the longer latency second wave. Notice how these diagonals appear that were not present before in the first wave. Right? First, second, first, second. And these diagonals correspond exactly to those pairs of images where the identities are the same, but there are different variations in the degradation type. And so these higher correlations along the diagonals mean that at longer latencies, which happens to be the time of the second wave, the neural population becomes invariant to occlusion and noise and just cares about the identity of the face. And this is exactly what's predicted by the generative feedback model. And here's ML during the first wave and during the second wave. So in each patch, we see increased invariance to degradation in the longer latency second wave compared to the first, consistent with generative feedback that's filling in missing information over time. Here are the average response time courses of ML and AM cells aligned to onset of the first wave. And not surprisingly, AM follows ML. And here are time courses of the same cells aligned to onset of the second wave. Now, surprisingly, the latency of activity in the two phase patches is almost the same. And you can see this in the raw single trial data, too. So here in the first wave, ML clearly precedes AM, while in the second wave, they have more or less the same latency. And this suggests that visual processing happens in two discrete phases. A feed-forward phase, followed by a recurrent feedback loop in which there's strong bidirectional coupling between different nodes of the processing hierarchy, leading to simultaneous responses. So during the feed-forward phase, the brain is humbly gathering vertical physical information, and so it distinguishes faces that are degraded in different ways. While during this recurrent feedback loop, we think it then interprets the data and fills in missing information to resolve ambiguity. So now let's turn to binocular rivalry. And this study was led by a postdoc in the lab, Johannes Hesse, who's truly extraordinary and brave, and he's also on the job market now. So bistable percepts, where the stimulus doesn't change, but your conscious perception of it flips back and forth, strongly suggests some kind of internal brain dynamics is involved in generating conscious visual perception. And we leveraged a particular bistable percept of binocular rivalry to study how conscious percepts are generated in the face patch system. So in rivalry, as I briefly mentioned before, we showed two different images to the left and right eyes, and then your conscious perception switches in an all or none way between the two images, even though the physical stimulus never changes. And this lets us study mechanisms for generating subjective experience that are independent of physical encoding. And we can tell which image the monkey is subjectively perceiving by training it to follow a fixation spot that moves differently in the two eyes. So if he follows the spot in the right eye here, then we know he's seeing the house. And if he follows the spot in the left eye, then we know that he's seeing the face. And in human subjects, we could correctly predict their button press reports of what they consciously perceived from their eye movements with 93% accuracy, confirming that the so-called no-report paradigm works. So in the 90s, Nikos Logothetis and colleagues trained monkeys to look at binocular rivalry displays and report their conscious percept with a joystick. And then they recorded across the visual system, and they found only low percentages of cells modulated by conscious perception in early visual areas, but very high percentages modulated by conscious perception IT cortex. And from this, they concluded that IT cortex represents pure conscious perception. Now we revisit this story with neuropixels, and it turns out to be more interesting. So here's a snippet of activity of cells from face patch ML when the monkey was looking at an unambiguous face alternating with an unambiguous object. And not surprisingly, face cells responded more when the monkey was looking at a face. Here's a snippet of activity from the same cells during binocular rivalry. And we inferred from the monkey's eye movements that his conscious percept was face in these red epochs and object during the blue epochs. So the population responded more during the red epochs when we inferred that the monkey consciously perceived a face, even though the physical stimulus remained constant throughout. And using population activity, we could correctly decode the monkey's percept on single trials with 88% accuracy. So far, the results seem consistent with the classic story. IT cortex represents conscious perception. But is that all there is to it? Is IT cortex really just representing pure conscious perception? To test this, we presented two different types of rivalry stimuli, made of either AB or AC. So now when you're conscious of A, the suppressed stimulus could be B or C. And if the neurons are truly representing just your conscious perception, then you shouldn't be able to decode B versus C. Here you can see the decoding accuracy for the suppressed stimulus as a function of the length of neural response interval used. So we found that at intervals of 1 second and longer, we could decode the suppressed stimulus with 100% accuracy. And the mean perceptual dominance duration was much longer, around 7 seconds. So this contradicts the classic story that IT cortex reflects pure conscious perception, because we could decode the suppressed stimulus perfectly on a 1 second basis, even though the monkey was not conscious of it. And so IT cortex represents both conscious perception and the suppressed stimulus. How is this possible? In theory, there are two ways that IT cortex could represent both the conscious and the suppressed stimulus. One hypothesis is that IT cortex alternates between representing conscious perception and the physical stimulus. And we call this alternating hypothesis the Baumkuchen hypothesis. And this can be contrasted with the chocolate pound cake hypothesis, where every bite of neural activity contains both chocolate and cake, or in this case, representation of both the conscious stimulus and the suppressed stimulus. And to distinguish these two hypotheses, we leverage the power of large-scale single neuron recordings, which allows us to decode what's represented on very short timescales. And first, as a sanity check, we want to know if we could successfully decode the physical stimulus using very short intervals of 100 milliseconds before looking at temporal dynamics during rivalry. And so here is a 5-second long snippet of activity of responses of over 700 simultaneously recorded neurons from face patches ML and AM during presentation of a physically unambiguous stimulus. So stimulus A is alternating with stimulus B. And now we slide a linear decoder trained on 100 millisecond intervals to decode A versus B. And you plot the decoder confidence, and you can see the decoder is certain that it's A when the stimulus is actually A, and certain it's not A when the stimulus is B. And the decoder confidence margin is high and stable during the entire period. And for the more anterior face patch AM, the decoder margins are very consistent. Now I'm going to show you activity of the same cells during binocular rivalry. So we know the monkey was consciously perceiving the face throughout this specific 5-second snippet based on his behavioral report. And later, his percept switches. So you can see from the raster that there's very interesting dynamics and coordinated changes in activity across the two patches. And this dynamics is amazing because the physical stimulus was not changing at all during this time period, and also the monkey's conscious percept was not changing at all. When you try to decode what the neurons are representing with a sliding window decoder, we find that the confidence for decoding the conscious percept is not constant, but instead it rapidly fluctuates. So there are the peaks when the decoder is very confident that the stimulus matches the conscious percept, but there are also all these valleys, and what's happening during these valleys? So here in blue is the curve showing confidence for decoding the suppressed stimulus. So we see during the valleys of the red curve, the cells are representing the physical stimulus, such that we can decode the unconscious stimulus that the monkey doesn't even perceive. And this interdigitated representation of conscious percept and physical stimulus was highly coordinated across different patches, which is amazing, again, because the stimulus was not changing at all. So the brain is representing what we consciously perceive in discrete epochs around a few hundred milliseconds long, interleaved with representation of the physical stimulus. And it's very clear that neurons in the face patches are not just representing what you consciously see, as the classic story developed by Nikos Logothetis had claimed. And to prove to you that I didn't just cherry pick that snippet that I just showed you, here you can see the correlation between the conscious and suppressed decoding curves across all 5-second intervals in the session, and it's highly negative. And this anti-correlation was also consistent across sessions and monkeys. So we have empirical evidence for the Baumkuchen hypothesis, but this is an extremely strong and surprising claim. So are we really sure? Next, I'm going to show you 2 analyses that really convinced us that this is real. So in the first analysis, I'll show you that during the unconscious physical epoch, you can actually discriminate the suppressed stimulus. So we're going to compare the fraction of time points where the conscious decoder is most confident with the fraction of time points where the conscious decoder is least confident. And for these 2 sets of time points, we'll look at how well the suppressed stimulus can be discriminated. And if Baumkuchen is correct, then the suppressed stimulus will not be discriminable on the left, but will be discriminable on the right. And this is exactly what we see. So we see that during the conscious epochs, for AB, the decoder margins for the suppressed stimulus are centered over 0 here. And for AC, the decoder margins are highly overlapping. So for these conscious epochs on the left, you cannot discriminate the suppressed stimulus. And now for the fraction time points where the conscious decoder is least confident about perceiving A, here on the right, we see that the suppressed stimulus becomes very separable. So this shows that it's generally true that during rivalry, there are pure conscious epochs where decoding of conscious perception is high, but the suppressed stimulus is not represented, and there are distinct epochs where the physical stimulus is represented rather than the conscious percept. And now I'm going to show you a second analysis that was critical for convincing us. And here we performed a shuffle analysis where for each cell, we shuffle the time points within each condition before applying the decoder. And so we shuffle for each cell independently, but only with other time points of that condition. And this checks if the Baum-Kuchen effect arises from precise coordination between cells driven by intrinsic brain dynamics, or if instead it's some kind of decoder artifact. So if Baum-Kuchen is real, then after destroying the coherence between cells by shuffling, the effect should go away. And this is exactly what we see. So the strong anti-correlation effect that we saw earlier completely goes away. And now for the shuffle data, we can decode the suppressed stimulus throughout, even when the conscious decoder is confident over on the left. So this is a strong control that the Baum-Kuchen effect is real. And so to recap, I just showed you that for the real neuropixels data, there was a strong, robust anti-correlation between confidence for decoding the conscious percept and the suppressed stimulus, consistent with the Baum-Kuchen hypothesis, while for the temporally shuffled data, this anti-correlation disappeared on the right. And together, these two analyses really confirm that the brain is coordinating switches in representation between conscious percepts and physical reality. And overall, we think this is pretty remarkable. It means that we have concrete neural evidence that consciousness is encoded in discrete epochs, spliced with frames where you're unconscious and your brain is just processing the physical stimulus. And Julian Jaynes intuited that consciousness is likely discontinuous, and he describes this beautifully in his seminal book on consciousness, where he writes, "Consciousness is a much smaller part of our mental life than we're conscious of, because we cannot be conscious of what we're not conscious of. How simple that is to say, how difficult to appreciate. It's like asking a flashlight in a dark room to search around for something that does not have any light shining upon it. The flashlight, since there's light in whatever direction it turns, would have to conclude that there's light everywhere. And so consciousness can seem to pervade all mentality when actually it does not." And I think anyone who's been under general anesthesia can attest to the truth of this. And instantly, Julian Jaynes was trying to establish that consciousness is not necessary for most cognitive functions, and so it's even possible to have a whole civilization without consciousness. So we think this pattern of results that I've shown you could be parsimoniously explained by the analysis by synthesis framework, where the physical stimulus is represented during feed-forward phases and the conscious percept is represented during the feedback passes, and the two are naturally interleaved. And now we're collaborating with Tatiana Engel to model the interaction between different face patches as a coupled dynamical system to look for direct evidence for generative feedback interactions between patches. And we still don't know what's ultimately triggering these two phases of activity and recording across many different higher brain areas, including prefrontal cortex and hippocampus, to try to find out. So far, with degraded faces and rivalry, we've been studying this generative pathway as part of a closed loop that has both feed-forward and feedback, because that's how vision normally operates. But can we somehow isolate the generative pathway to get direct evidence for its existence? And this is what we tried to do next. In a study of human mental imagery that was done in collaboration with Uli Rudeshauser at Cedars-Sinai Hospital in Caltech by our joint graduate student, Varun Wadia, who's now a postdoc with Uli. So Uli's lab records from human epilepsy patients who have electrodes implanted throughout the brain for the purpose of finding the source of epileptic seizure. And so we recorded neural activity from single cells in ventral temporal cortex of these patients while they observed pictures of different objects. And we then asked them to imagine the same objects, triggered by verbal cue. And first we asked what the tuning looks like for visually perceived objects in human IT cortex. And it turns out that the human IT cells basically use the exact same axis coding scheme as macaque IT cells, where each cell shows increasing response along its preferred axis and no change along the orthogonal plane. So here are the most and least effective real object stimuli for one example cell. And by presenting many objects to the cell, we could map its preferred axis. And then knowing its preferred axis, we generated super stimuli that had even higher or lower projection onto the preferred axis. And we confirmed that these super stimuli could elicit stronger responses in the cell than any real stimulus, which you can see by comparing the color of the dots along the cross, which represents responses to the artificial stimuli, with the other dots, which represent responses to images of real objects. And using this access code, we could also reconstruct objects using responses from a small number of access-tuned human IT neurons. So the million-dollar question is, how do these human IT cells, which respond exactly like macaque IT cells during visual perception, respond during visual imagery? Here you can see visual responses to 8 different objects from one neuron in human ventral temporal cortex. And this neuron responds most strongly to this picture of a laptop. And here's the response of the same neuron during imagination. So it responded most strongly to the imagined picture of the laptop, even when there was no visual stimulus present at all. Here's another neuron. This neuron responded best to the piano. And here's the response of this neuron during pure mental imagery. Again, it liked the mental image of a piano. Interesting, there were also cells that responded only during mental imagery and not all during visual perception. And we don't understand these cells yet. So overall, across the population, a substantial 40% of access-tuned cells in human ventral temporal cortex, which is traditionally considered pure visual area, were activated during mental imagery. And here's the distribution of the correlations between the responses to real and imagined images in single cells. And so you can see the high correlation values indicate that most of the neurons reactivate in a way that respects their visual code. And I think this is the strongest direct evidence we have so far for the existence of a generative model in the primate brain. So finally, I want to sum up and tell you a little bit about our future directions. So to summarize overall what I've told you today about the brain's generative model for reality, the analysis by synthesis framework proposes that clear, self-consistent conscious percepts are created by a generative model via feedback. And in support of this, we found that face cells use a shape appearance access code to represent faces that is parsimonious and supports efficient face generation. During degraded face perception, face patches sometimes show a coordinated second wave of activity, which may correspond to ambiguity resolving recurrent feedback. During rivalry, the brain switches rapidly between states of physical processing versus conscious perception, which may correspond to feed-forward versus recurrent feedback. And during mental imagery, the brain generates visual representations that are consistent with feed-forward representations. So my ultimate goal is to understand how a monkey perceives the real world. And this, of course, goes way beyond understanding what's happening in the brain during passive viewing of flashed images of isolated objects, to understand what's happening during active exploration of different environments, as we see here. And so to this end, we're now recording from monkeys exploring 3D VR environments while searching for different faces. And the monkeys love playing this game, as you can see, and recording across the entire brain with neuropixels probes to understand how this dynamic spatial environment full of objects is represented. And this VR environment generates a whole slew of exciting new questions about how the brain performs multi-object segmentation and tracking to generate a stable representation of the environment despite drastic changes in the sensory input. I hope these experiments will give us a deeper insight into the fundamental data structure that the brain uses to represent the environment around us. And maybe also connect our work over the last 2 decades on face processing with my original dream to understand 3D vision. And so with that, I'll conclude, and I wish a very happy 20th anniversary to the Kavli Institute. Thank you.
So, brilliant research and a wonderful talk. 2 quick questions. First, you showed at the end that the neurons in the infratemporal that are active when you see an object are also there when you're trying to imagine it. What about V1?
Yeah, I don't know. I mean, human neuroimaging suggests that V1 may also be activated, although there was like a recent paper that said that in aphantasics, they also have V1 activation, so it's not sufficient for this mental imagery. I mean, I don't know. Our electrodes were in the temporal lobe where these seizures usually occur. So I think there are ways to get a monkey to also imagine faces, so I think probably we'd have to go that route.
Okay. Second quick question. At the beginning, you talked about these features, which are either shape or appearance. There were 50 of them. How did you pick them?
This was really like, we took this computer vision model. Stephen found this model, and so you do PCA, so it's completely unsupervised, right? You just align all the landmarks and you do PCA on the landmark locations and the texture maps. So that's how we pick them. Is that what you mean?
Yeah, that's right. So you use PCA to pick
Yeah,
them.
yeah,
Okay,
yeah.
great.
And I should say, like, it might seem like we pulled a rabbit out of the hat. Like the shape appearance model, we took it, and it just happens to explain the face patch responses. But I think there's a deep reason for that, which is, why did the computer vision people come up with this model in the first place? And it was in order to track faces, invariant to like the change in view and so on, which is, I think, what the brain also wants to do, and that's why it's generating this factorized representation of shape and appearance. Yeah.
Haraj?
Thank you very much for the wonderful and thoughtful talk. So my question is, if you go back to your... Actually, it was your turn. Sorry for...
Yes, there are two microphones I'm seeing. Sorry.
So if you go to your schema of these arrows going from early representation to the high-level representation, then coming back all the way down, one could argue that you need to go all the way up because that's where the big picture is. And now you kind of have to create your hypothesis about the world and come back and regenerate it.
Yeah.
The other view is that this is a scalable process. Even in V1, there is a little loop. And then from V2 to V1, there is a little loop. And then V4. How do you think we can discriminate those two and how different they are? Does it really, are we forced to think that it should go all the way and then come back down? There is an argument for that because that's where the big picture is. Or you think it's a scalable process that means has a meaning at every scale, basically?
Yeah, that's a wonderful question. I mean, I can say that I think we know that it does go all the way up. So your question is, does it have to go all the way up? So the evidence that we know that it does go all the way up comes from this beautiful experiment from Yasushi Miyashita, right, where he cut the posterior corpus callosum and, presented stimuli to the left visual field, and then it would go to the right hemisphere. And so it had to go all the way up and cross the anterior commissure before coming back down to where his electrode was, and it was able to do that, right? So we know it does go all the way up in the real brain. Does it have to do that? I suspect not. Just the way the circuit is wired, like it seems hard for me to imagine that these feedback connections, required to go all the way up, right? They should, it seems, be able to provide the feedback, without getting the signal from the higher level area. I don't know of experimental evidence that's true. I think it'd be really hard to test.
I guess one way to put it is that is it a global, is Baum-Kuchen model a global model, or is it a high level visual?
Yeah, I think my intuition is that it is a global model. I think that, for example, with neglect, that there are like specific processes that just completely knock out your visual perception. It's not like, just incremental all the way up. So that's my intuition. I think the parietal cortex also plays a very big role in representing this big picture that you're talking about, like a scene graph of all the objects around you. So I think we need to go hunt there. So I think it's not just, like you say, scalable. I think there is modularity, and there are specific areas that are representing the global picture that are essential.
Thank you.
Okay, so restoring the order here, we'll go Kay, Ralph, and the back microphone, and then we'll bounce back and forth. I think that's the order that people came to the microphone.
Sorry, we're so excited. We're all fighting to ask questions for Doris. This is an amazing talk. So really loved your talk. And then my question is actually about the stimulus set and the parameters for this last experiment, which I thought was so cool about the mental imagery. And then you just almost did it as a throwaway slide that you just speeded through, saying that sometimes there's neurons that respond to the mental imagery without the visual response. But then you didn't really talk about that particular stimulus, but it looked like maybe a pair of tigers, and then the actual image wasn't very dynamic. But if I were to imagine a pair of tigers, I could create a visual image that was greater. And it started me wondering about how you parameterize the worthiness of the words matching to the visual stimulus, if that makes any sense. Like, you can imagine lots of different visual images that are a pair of tigers walking, and then, within that set that fits the verbal cue, how do you deal with that dimensionality?
Yeah, so in this experiment, Varun just picked like a pair of images during each trial, the imagery trials, right? So it was very clear. It was like, a person or a coffee cup, and then the person would see those images, so he had a period to learn those, and then it would just be this verbal cue where he had to, visualize A, B. So I think the ambiguity in how you describe it wasn't really, an issue, I think, here, because it was just two images that the person had to select between with the short-term memory.
Hi, thank you. Really cool talk and amazing data. I have two questions that are connected. One is on the switches between the conscious percept and the representation of the physical stimulus. What's the distribution over those switching times? Is that more periodic? Is it more like stochastic, like from a sampling process?
Yeah, it's not a perfect oscillation. I think that the mean duration is like 200 milliseconds. Yeah, I should have plotted that, yeah.
But it is more regular than you would expect from like a gamma distribution or something.
I see. I don't know, actually. Yeah, we should look into that.
Okay, cool. And do you see any evidence of those oscillations during the mental imagery as well?
I don't think Varun has looked. That's a very interesting idea. Yeah.
Yeah, so your model didn't have any nonlinear components, if I understood correctly. Yeah, back here. Is that right? So your model was totally linear, if I followed.
Which model are you talking about?
Well, I think that the generative, the matrix model that you were introduced.
Oh, the shape appearance model. Yeah, so all the nonlinearities are absorbed in the parametrization, right? The transform between the base pixels and the shape appearance parameters. And then what you find is that the brain encodes these shape appearance parameters linearly. Yeah.
Okay, that was my question. So there are nonlinear elements. They're
Yes,
okay.
It's highly nonlinear, yeah.
Okay, very good.
Stuart Anstis.
Hello. In 1910, Perky asked her observers to form the mental image of a bunch of bananas on the wall. She then surreptitiously turned up a projected physical picture of the bunch of bananas, and observers mistook this physical picture for a mental image. Is this consistent with your story, and do you think you could replicate that experiment?
Yeah, I'm very suspicious of that result. I mean, I'm introspecting. I don't think that would ever happen to me. Like you're saying that I would form a mental image, and then someone would show me a picture, and I would look at the picture, and I would think that was my mental image. I don't know. That seems strange to me. And I don't think the results are... I think it's very easy to distinguish, for example, between the representation from the mental image versus the physical image, right? Only 40% of the cells were reactivated by the mental image.
Thank you.
Yep.
I'll ask a quick question. So you started your talk by describing this network of 6 face areas, all interconnected. So it's sort of tantalizing to think about how they interact with one another in the construction of a face. Are there aspects of face processing that occur earlier than others? And do those show up as occurring earlier in different patches? And what can you say about the dynamics of the interactions between the patches?
Yeah. I mean, just so Vinreg, I found that there's increasing invariance as you go more anterior. So the posterior patches represent faces at different views. I think Elias Issa recorded even more posterior in face patch PL, which is between V4 and TEO, so very, very posterior. And you find cells that are tuned to the contralateral eye there. And then in the most anterior patch, the hallmark is really this extraordinary invariance, right? Not just to view, but also if you there's this phenomenon where you can take a face and you can squish it, you can change the aspect ratio to be 10%, and you still have no problem recognizing it. And these cells also reflect that. So definitely there's a functional hierarchy going anterior, and that's all in the feed-forward response. So what the interactions between the face patches, of course, the reason why we went to all this effort to try to record simultaneously from them. So far, I can't say that we've had a lot of insight into that, right? We found that, in the degraded faces, the second wave of response, the timing was simultaneous, but, what does that mean? We don't know. For rivalry, as I mentioned, we're collaborating with Tatiana Engel to try to understand better, when you have a feed-forward versus, or a physical versus a conscious epoch, is there a difference in the dynamics between the interaction? So far, just looking at the latency distributions, we don't see anything clear. We're hoping that her analysis might pull something up. So, yeah, this is something for the future.
All right. I think Terry is back with us.
Well, I want to follow up with Stuart Anstis' comment. I think that there are cases where you do generate things from noise. For example, I see faces sometimes on tree bark. Even Shakespeare has Polonius seeing images in clouds, right? So if you have just noise, you can pick things out that from your generative thinking.
Agree completely. And we're studying this now with Mooney faces, like, well, what happens at that moment of recognition? So we have monkeys like looking at Mooney
wonderful.
faces, and they have to touch on a screen the eyes, and so we can tell when they actually see the Mooney face and see what's different about the dynamic. So I completely agree with you there. I was just saying, like, I don't know, if imagine something and you look at they show like a real stimulus and you think it's your imagination. I feel like that's different from seeing a face in clouds.
No, it's different, but there must be, between those extremes, there must be some kind of a continuum, right?
Mm-hmm. Okay.
In other words, where you can,
Yeah.
I sometimes mistake people I know from, some stranger just because there's a similarity, but, it's clearly different.
Yeah, and of course, dreams are this instance where we generate very vivid percepts purely internally. And, I'm constantly trying to find a student who wants to come and record from sleeping monkeys at night.
Hi, thank you for that wonderful talk. My question relates to the stimulus images that you used. I noticed you had all human faces. What would you expect to change if you were to show different macaque faces or familiars or things that were interpretable by the monkey, other than a picture of a house, which presumably it would not have seen?
Yeah, we have shown monkey faces. In fact, we generated a parametric shape appearance space defined completely by monkey faces, right? So we could show 2,000 monkey faces. I didn't talk about that because the principles were essentially the same. They showed this ramp shape tuning to these different shape appearance axes also for monkey faces. With respect to meaning, we have found that the representation of familiar faces is totally different at long latency, and this is super interesting, right? So if you look at the coding axis for unfamiliar faces, it stays the same over time. And if you look at the coding axis for familiar faces, initially it matches that for unfamiliar faces, and then it totally changes. And I think that's, it reflects some kind of lateral organization in the cortex, right? That's, pulling out the context, the memory, and it's changing the representation. So there definitely is representation of meaning in IT cortex. Like many people say that IT cortex is just representing shape. And I think that's true of the initial feed-forward component. I think that's not at all true of this long latency component.
We have time for just one short question.
Okay, thank you for the talk. Very cool work. Mine is actually just asking for a comment about what do you think is coming next? Like what would you, so with all the generative AI technologies that we have now, and then this talk about, and the whole symposium about the brain and being generative, what would you say is coming for the next 10, 20, 30 years?
Yeah, so I think the most exciting thing is that I think AI is actually stuck right now. I think, what is the next big thing? They're running out of training data, and I think the next big innovation is going to come from us neuroscientists. We're going to figure out the secret to AGI, and we're going to figure it out because, our brains can run on 30 watts, right? And we're constantly generating images, whereas, OpenAI, like Sora, they have to go out to lunch to wait for it to generate one video because it requires so many GPUs. So I think neuroscience is going to discover what are the structured priors that you need to build an efficient intelligence. And I think these experiments exploring representation of natural environments, animals navigating these natural environments, and recording all over the brain with neuropixels probes, I think those experiments, have the potential to reveal those new principles that can drive the next innovations in AI.
On that note, let's thank our speaker, Doris, for a wonderful talk.